Today we're beginning Unit 4, Genetics. This is a fun unit to teach. It's a fun unit for my students as well. And we're going to start with kind of a historical walkthrough of the history of classical genetics from Gregor Mendel all the way through molecular genetics and what we're currently working on, geneticists are currently working on. Of course, right now, we are all interested in COVID and the coronavirus. And geneticists are really some of the frontline scientists working to figure out what this virus is and in different ways that we can prevent it or we can treat the disease. And it often starts by understanding the virus at a genetic level. And there are scientists that are out there collecting samples from different regions and sequencing the genome. And some of the scientists working at UCSF down in San Francisco, these are scientists that worked on the last SARS outbreak about 20 years ago. And they were reporting that when they were sequencing from the SARS-CoV-1 virus, it was about $10,000 per genome to be sequenced. And now the cost has gone down, the technology has improved, and it's about 1 to 10 cents for every genome that they want to sequence now. And so the ability of scientists now to collect samples from the environment from different patients and to see how this virus is mutating and changing and passing that information on to the protein biochemists that can create constructs of these viral proteins to understand how the virus enters cells and to figure out ways that you could possibly prevent the virus from entering cells. It often begins with genetic science. And I think it's important to take this historical view from Gregor Mendel all the way into now to appreciate how far we've come and how quickly we've made some changes. And also this is a point at which I think students get excited to say, what could I do with this information, with a degree in biology? How can I be a part of this process of understanding and being able to contribute to a current research topic? I'm also interested in genetics from a purely family point of view. And I think that we are all created in the image of God, and we're all related to our families. And when we look at our families in the photo albums, we see similarities to one parent or maybe to the other, or maybe to neither parent. And but we have a great grandfather's physical characteristics or something. And so the questions of how our physical appearances are passed down, that was a mystery for so many, many years. And to understand classical genetics and where we started to understand the pieces, put the pieces together is fascinating. My dad met my mom in college, and they met through my mom's brother. So my dad and my uncle were roommates at Seattle Pacific, and my dad is 5'2", and my uncle is 7 feet tall. And if you don't have the spatial perspective, that's a big difference. That's a really big difference in height. And the best part about that relationship is that they were partners on the tennis team. And I just think that would be like this dynamic duo that would be terrifying to play against. Anyways, that's how my dad met my mom's family, and then the rest is history. And here I am, a healthy 5'6", but I did not get my uncle's height. So the study of genetics tells us why we are similar, and it tells us why we are different. And it also tells us about how not only we can adapt to our environment, but how our genetics can change and evolve over time. And all of these stories are woven together in this unit, I think, in fascinating ways. It was about 140 years ago when scientists were starting to use microscopes to watch cells go through mitosis and meiosis and divide, and they were watching what they didn't know were chromosomes being divided between the two cells. And it led to what was called this particulate theory of inheritance, that there was something physical being passed down from one cell to another as a cell divides. And Gregor Mendel was working with this garden piece in about this time in this space, not looking through microscopes, but planting peas and watching the passing down of inherited traits. And we'll start our discussion there looking at these two laws of inheritance that Gregor Mendel proposed. and as a beginning for this conversation of how you can do crosses with different types of species and you can eventually predict the outcomes. And so we'll also look at how probability and how mathematics and statistics goes into this model of prediction what you might see. And this is a really important place in the history of science. It is now not a mystery of what can be expected after organisms reproduce. You can actually control that. And of course we knew artificial selection, we were selectively breeding crops or breeding different sheep or different organisms for their traits. But as we learn more and more about how the inheritance patterns work, we can now make predictions. And we use these predictions today in genetic counseling because the Mendelian traits that he observed in peas, we can observe now in human characteristics, and many human diseases, for example, follow Mendelian patterns of inheritance. In chapter 15, we'll now start with a scientist named Thomas Hunt Morgan and his work with fruit flies, Drosophila. And the importance of this work was to now pinpoint genes onto chromosomes. And so now we're knowing that it is DNA that is being passed down from one cell to another cell, and that there are regions on these chromosomes where these genes are located. And these genes are giving rise to the traits that we're now following. So Thomas Hunt Morgan worked with fruit flies, and we'll look at his work with fruit flies for two important reasons. One is we'll look at a type of inheritance called sex-linked inheritance. And these are when genes are located on either the X chromosome or the Y chromosomes, the sex chromosomes in flies, the sex chromosomes in humans. And they have different inheritance patterns than the Mendelian genetics that we saw in the previous chapter. The second important part of chapter 15 is to physically map the location of genes onto chromosomes. And you can do this with a series of very ingenious breeding experiments where you cross traits on genes on what we call our linked genes. And linked genes are genes that are found on the same chromosome. But by looking at the results of these crosses, we can now physically put those genes into physical places onto chromosomes and map the locations of chromosomes. And this is all amazing science for 100 years ago. And of course, now we don't need to do any of this because now we can sequence a gene, a chromosome, and we can know exactly where on that chromosome a specific gene is by sequencing a genome, for example. As we work through the history of genetics, in chapter 16, we now get into a very critical time, and that is the discovery of DNA and the structure of DNA. And we'll talk a little bit about the story of that discovery and how that discovery was made possible. And we'll talk about the implications of what it means for molecular science going forward after we have that structure. But the beauty of the discovery of DNA was a molecule that predicted its own function. You could look at it and you could tell exactly from the way that DNA is constructed how this molecule can be replicated and how this molecule can be used for gene expression, for the process of transcription. And you can look at this molecule and see how stable it is and how it is used as the blueprints of that specific species and how it can be stable over the history of generations, as well as being able to be manipulated, the ability to be changed, the ability to have random mutations that affect the structures and then the function of the genes. And so understanding this structure and the importance of this discovery forever changed the world of science. And I think every aspect of science, whether you're studying ecology, whether you're studying in population genetics, whether you're studying developmental biology, all of these sciences now have a genetic and molecular component because we realize the importance of how we can manipulate genomes now and understand basic scientific questions at this level. Finally, at the end of this unit, in chapter 17 and 18, we're going to look at the process of gene expression. So this is the process of transcription and translation in chapter 17. And then in chapter 18, we'll be talking about how this process is regulated. And we'll look back at the differences between prokaryotic gene expression and eukaryotic gene expression and how these processes are similar and how these processes are different. One interesting difference in prokaryotic gene expression is that the genes in a bacterium, there aren't as many of them, and they're often all located together in a cluster called an operon. And this allows multiple genes to be controlled by one promoter and what is called one operator. We can turn on a whole set of genes or turn off a whole set of genes as a function of responding to an environment. As the bacteria finds itself with food, it turns on a whole set of genes that are able to produce enzymes that can break down that food. And then when it does not have that food, it turns the whole set off.